Now I wanted to introduce you very interesting idea of one bead, one molecule library concept. So I think among chemical library, this is the most challenging and most interesting other idea, I believe. Let's think about peptide library. If you wanted to make peptide library as a mixture, you can prepare in reaction the vessel. This is resins, and you can add 20 different amino acids. For simplicity, I just used the 3, but let's assume that you added 20 amino acids here. Then first amino acid you can load onto the resin, and N-terminal is the protected, so that's why it can react only one side. And then you remove those protecting group, and then add second amino acid. Then you can make a dimer, and again you remove protecting groups, and you can add the third amino acid, and you can add, and you can purify. So this the resin bound the forms. If you had the trimer, already you have 8,000 other library. 8,000 is not bad, and if you clip them off, you have a mixtures of these peptide libraries. It's a mixture, it's a little problem. And if you look at the one bead, so one bead has many functional groups. That's why if you read this amino acid, then even one bead you have many different mixtures like this. If you generate 8,000 library, then on one bead also you can get 8,000 different peptide can be generated. So if you clip off, then it's the same situation, mixture of a different peptide. Because this is a mixture, it's now a little difficult to screen or utilize. So many cases you may want to have purified each different the library member as separate sources. That may be better. So mixture library, it's very easy to make, and just you add different amino acids at the same time, and you can make many molecules very quickly. But limitation is you can get only mixture of those peptide. Now screening can be a little difficult. Another approach we can call parallel synthesis. Now again, to simplify the picture, I just put only 3, but you can assume 20 different reaction vessels here. So 1, 2, 3, 4, and 20 reaction vessels here. And then you can do the reaction one step and move to next step and third step, and you can get this compound. So in this case, good thing is for each reaction path, you can get only one kind of peptide. That's really good. But how about the reaction number? Your final compound is 8,000. Then minimum you should do 8,000 reactions. And if it is 3 steps, that's 3 times of 8,000 reactions. Then you can get this tripeptide library, isn't it? So that's huge amount of different reactions you need to do. That's why practically you cannot make really huge library. Keeping this in mind, let's think about another possibilities. Now let's think about a totally new approach. So in this approach, in the beginning you have a total resin in one pool. And then again, to simplify, I just put only 3 reactors here, but you can assume you have 20 different reaction vessels for 20 different amino acids. Now these resins can be distributed to each reactor, even amount. And then each reactor has only amino acid A, B, C, so it can be reacted like this. Other leftovers can be washed out, and these resins can come back to the common pool and mix them very well. Now the resins can go to split again to different reaction vessels and go back. Now in this case, original resins are all mixed around, so A, B, C up to 20 different amino acids evenly distributed, we can think, and then wash out again, and they can pool again. Now we repeat this pull and split repeatedly. So if you do one more time, we can send back, and then they can be added other amino acids. But each reaction vessel, we don't use mixtures. We use only A, only B, only C. So only one kind of reaction can happen for one resin. After the reaction is done, if you wash out all the leftover the solution phase reagent, now end product is this resin beads. And each resins, you remember, when they split and they reacted only with one amino acid and they coming back to pull and then redistribute it and then do the second step reaction and third step reactions. For each resins, they followed only one type of reactions. That's why if you look at those single bead, and single bead has a multiple reaction site, but actually all those reaction sites pass the same history. That's why one bead carries only one peptide product. So this is the concept of one bead, one molecule approach. In this case, one bead can represent one identity of one library. So if the total resin number is more than 3.2 million, then even though you make a pentamer of your peptide library, now practically you have 3.2 millions of isolated library set. If you cleave off this peptide into solutions, if you mix them together, you lose your advantages. Now this is the same end result as of the mixture synthesis, isn't it? In mixture case, synthesis was quite easy. You just add 20 mixtures of amino acid and you generate a library. At the end, you cleave off and release everything into one pool. Now you have a 3.2 million of product in your final the reaction pool. That's a huge numbers, but it's quite difficult to test. So using this split and pool, you have a 3.2 million of unique resins. And you don't want to cleave off and then mix them together. You want to keep those resins separately and utilizing those resin, and you want to screen. Then you have very unique 3 millions of different resins for screening setup. Because of this property, you need to keep those compounds on bead and then test those library on bead before you cleave off. So it can be under magnifier, or you can use the microscope to visualize those bead. And those bead size, I said, 100 micrometer or 200 micrometer size. Sometimes it can be used for color generation, or sometimes you can use fluorescence. But your total numbers of bead is almost a million. Now you cannot just look at 10 by 10, so you need to look at minimum hundreds or thousands of resins like this format. And in this case, let's just say some color development is your screening, and maybe you can easily see now this one is definitely your hip. It's very strong, but many of them is kind of a medium color changes like this. Now you need to think what kind of criteria you should apply. So totally here is less than 1000 bead, you can get only one. But still, if your total numbers of resin is a million, still you may get thousands of this kind of a strong one. You should think what kind of algorithm, what kind of criteria you should apply. Now another the key question here is, all of this the color changing the method can be either enzyme reaction or binding affinity to small molecules or molecular level of event. If you want to apply this one bead one molecule library in cell screening, now we encounter different kind of problems. Now let's say this is our cells. Do you remember what was the cell size? Mammalian cells usually is a 10 micrometer or 20 micrometer size. How big was our resin for synthesis? The resins were much bigger. It's the 100 micrometer or 200 micrometer size. And then your active molecules or active library peptides are on the surface of this resin. Then if you want to test this peptide inside the cell, wow, this is too big to go inside the cell here. Maybe your screening should be opposite way. Maybe the cells should recognize, maybe some of those cell surface receptors recognize this peptide and then they prepare to bind to this peptide. Can be one kind of assay you can think. Size-wise, this is kind of limitation of your screening. And this kind of a binding assay of cells to the resin is possible? Well, seems like it's possible. So this is the resins, and then you can see here, some of those resins has a much preferred than the other resins by the cells. The cells recognize this the surface, and then the cells bind much more preferably on this resin than other resins. So in this case, now the next question is, if you pick up this resin, then can you sequence the peptide from one of this single bead? The question is, the peptide on this one resin, is that enough amount to get out the identity of the peptide or not? So to get some practical other numbers for that, using this example, let's think. So read this other question carefully, and this question will be the IBLE. So think about the answer and then put up your answer on IBLE. Now utilizing the one bead one library approach. Now if you want to extend your library size, now the millions is quite big amount. So there is some other tricks to better classify your library. So let's say if you want to make the peptide library as a 5 mer, 6 mer, or 7 mer, 8 mer, 9 mer, different size, and you want to test them all. One trick can be you can add different colors of the tagging system into resin. Now you can look at your resins by looking at those resin colors. If it's a red color, then you can say, oh, this is 8 mer. Then it can help potentially later to, after you take out your resin and then analyze those peptide sequence, then you can further narrow down. Okay, I expect only 8 mer. It may help you for further analysis. Even though you have different colors here, now again, similar type of the assays, this resin has much more cells than the others. So the peptide found on this resin has stronger affinity to the cells, we can think.